Types of microscopes. Microscopy is the study of objects too small to be visible to the naked eye. Different types of microscopes enable various levels of magnification and can be classified according to the way they create images from samples. The two main types of microscopes are optical and electron microscopes, which use light and electron beams, respectively, to illuminate specimens. The light, or optical, microscope was invented in the 17th century and remains essentially the same, though it has been refined in many ways over the years. It employs visible light and a system of lenses to magnify images of small samples. The simplest light microscope consists of an objective lens and an eyepiece maintained at an up and down in relation to the sample. It works by forming on the objective lens a real intermediate image, which is then greatly magnified by the eyepiece. The two most common types of light microscopes are compound microscopes and stereo microscopes. Stereo microscopes are frequently used to observe larger, opaque species since they generally do not magnify as much as compound microscopes, around 40 times to 70 times maximum. Yet, they can give a truly stereoscopic view because the image delivered to each eye is slightly different. Compound microscopes, on the other hand, can magnify up to about 1,000 times, yet are not capable of producing a 3D view, even if they possess two eyepieces. While stereo microscopes do not necessarily require elaborate sample preparation, the specimen used in a compound microscope has to be sufficiently thin and bright for the microscope light to pass through. A light microscope, even one with perfect lenses and perfect illumination, simply cannot be used to distinguish objects that are smaller than half the wavelength of light, around 0.275 micrometers. To see tiny particles under a microscope, scientists must bypass light altogether and use a different sort of illumination, one with a shorter wavelength. The introduction of the electron microscope in the 1930s filled the bill. In this kind of microscope, electrons are sped up in a vacuum until their wavelength is extremely short, only 100,000th that of white light. Beams of these fast-moving electrons are focused on a cell sample and are absorbed or scattered by the cell's parts so as to form an image on an electron-sensitive photographic plate. There are two different types of electron microscopes: transmission electron microscopes, TEM, and scanning electron microscopes, SEM. TEMs passed electron beams through specimens, allowing their viewer to see structures within a specimen. SEMs are less powerful, providing less magnification, but are best for examining the surface of a material. Basically, SEMs show topography of a sample, while TEMs show internal detail of a sample. Generally, electron microscopes are better than its optical counterpart for a couple of reasons. The first reason is that electron microscopes use electrons with wavelengths much shorter than that of photons, the basic unit of light, to magnify a projected image. Therefore, they are able to achieve unprecedented magnification of up to 1 million times compared to the limited magnifying power of optical microscopes of only 1,000 times. Aside from this wide difference in magnification capability, electron microscopes are also able to project a more detailed field compared to the fuzzy details of the optical microscope, allowing for the visualization of structures that would normally be not visible by optical microscopy. For all of these advantages, electron microscopes cannot surpass optical microscopes in one aspect: the ability to observe living cells. With optical microscopes, it is possible to observe a wide range of biological activity, such as the uptake of food, cell division, and movement, or even the uptake of colored pigments by the cells with the additional aid of in vivo staining techniques. These processes cannot be observed in real time using electron microscopes, as the specimen has to be fixed and completely dehydrated, and is therefore dead. Besides, the low cost of optical microscopes make them useful in a wide range of different areas, such as education, the medical sector, or for hobbyists. Generally, optical and electron microscopes have different areas of application, and they complement each other.